Charisma Therapeutics and some individual contributors. So thank you very much, everyone, for your support and for participating. So our co-founders for this year are Jennifer Guerriero, myself, Dimitri, and Jelani Zarif. So we will be your hosts for this series this year and in the coming years. So we look forward to sharing these wonderful seminars with you and hopefully organizing additional meetings as we had last year. So I think I'll wait just one more minute for our guests to come in. They usually come in right at the hour. So. Okay, well, it gives me great pleasure to introduce our speaker for today. Marco, you could share your slides if you like. All right, so our speaker today is Marco Colonna from Washington University in St. Louis. Marco was born in Parma, Italy, received his medical degree and specialization in internal medicine at Parma University, and then performed postdoctoral research training at Harvard Medical School. He was then a member of the Basel Institute for Immunology, and since 2001, he's been a professor of pathology and immunology at Washington University in St. Louis. Since 2019, he's been a member of the National Academy of Science. His research is focused on immunoreceptors, so he has identified and characterized the killer cell Ig-like receptors and HLA-C polymorphisms as their inhibitory ligands, as well as the discovery of the LILR and TREM inhibitory and activating receptor families. So he's identified plasmacytoid dendritic cells as a source of interferon alpha and beta and antiviral responses, and innate lymphoid cells that produce IL-22 in mucosa. His current research includes innate lymphoid cells in mucosal immunity, plasmacytoid dendritic cells in host defense and autoimmunity, and TREM2 and innate immunoreceptors in Alzheimer's disease and cancer. So today he'll tell us about TREM2 and LILR receptors and neurodegeneration and cancer. Welcome, Marco.
Thank you, Julie. Thank you very much to everybody in the Myeloid network. I'd like to thank Jelani, Mitri, Jennifer, and Julie for this. I'm really delighted to have the opportunity to present our work to this forum. So today I'd like to review some of our data on TREM receptors and LIR myeloid receptor and show you some new data that are coming up from our lab. Let me just, I think you can see the screen well, I suppose.
Yes, so, it looks great.
Okay, good. So, well, one of the major interests in the lab recently has become the neurodegeneration, and particularly Alzheimer's disease, which is a major form of dementia that affects all people. The frequency of this disease increases dramatically with age and has become a major medical problem with the aging of the population. Now, the culprit of this disease is the generation of amyloid plaques from a protein called amyloid precursor protein, or APP, that can be cleaved by different proteases. And two proteases, BACE1 and gamma secretase, will then lead to the generation of a peptide called A beta peptide, which is prone to aggregation. And this peptide will then form amyloid plaques. So according to the amyloid hypothesis, the first event in Alzheimer's disease is the generation of these extracellular plaques that you can see here. And these plaques then induce neuronal stress, stress neurons then hyperphosphorylate a protein called tau, which is associated with microtubules. And this hyperphosphorylated tau tend to aggregate, precipitate, form what we call neurofibrillary tangle inside the neurons, which will cause eventually neuronal cell death and cognitive impairment. Now, this hypothesis is being supported by genetic studies on familiar Alzheimer's disease. These are rare forms of Alzheimer's disease that occur very early on during the life of these families. In fact, those mutations of APP and mutation of gamma secretase that facilitate the generation of a beta peptide and cause Alzheimer's disease. But when geneticists look at the vast majority of people with Alzheimer's disease, this is sporadic Alzheimer's disease, which represents more than 90% of cases of Alzheimer's disease, they found a number of different DNA polymorphisms associated with the risk to develop Alzheimer's disease. And we and others noticed that many of these risk factors actually affect genes that are expressed in myeloid cells, and particularly in microglia. So to this audience, I don't need to say much about microglia. I just want to remind you that these brain-resident macrophages have a unique morphology. They have this ramified morphology that was discovered by Del Rio Ortega. And they also have a unique development. They develop early on from a yolk sac, and they populate the developing brain. After the development of the brain is completed, then microglia will continue to proliferate and cell renew to maintain a population towards the end of a life. So we know for a long time that one of the functions of microglia is to control homeostasis, and particularly they have this very ramified morphology that allows them to control different areas or different sections of the brain. These were two photon images that were presented several years ago by Nimmerjan and colleagues. And you can see that the processes move constantly into these areas and survey the region. But then when we induce a lesion, for example, with a laser-induced lesion, you see the microglia send the processes to the lesion, try to contain the damage. And that's exactly what we see in Alzheimer's disease. So this is the section of a normal brain in which you can see microglia with the processes. And this is a section of a brain with Alzheimer's disease. You can see the plaques here, which contains A beta peptide, but also contain ApoE. And then around the plaques, you can see this microglia, the send the processes towards the lesion and try to maintain, to contain the spreading of the plaque. So we're particularly interested in this process because we know that one of the risk factors for Alzheimer's disease is the mutation of a gene called TREM2 that we cloned several years ago. In fact, TREM2 is part of the family of receptors that we clone. There are human chromosome 6, mouse chromosome 17. They mostly expressed in myeloid cells, although some of these members of this family, for example, NCR2, is expressed on innate lymphoid cells. TREM2, as many of other members of this family, is an activating receptor. It has specificity for phospholipids, apoptotic cells that expose phospholipids, lipoprotein that expose phospholipids as well. It's expressed in microglia, but it's also expressed in brain, in tissue macrophages, and is associated with a signaling molecule called DAP12, which is important for both recruitment of TREM2 on the cell membrane and also for signaling. So based on this association with Alzheimer's disease, we decided to see whether TREM2 is important to mediate microglia responses to A beta plaques. And so we crossed our knockout mice with a model of amyloid plaques called the 5xFAD transgenic models. This is a transgenic mouse that expressed human APP and human gamma secretases with 5 different mutations that facilitate the generation of A beta. And so what we saw was that in a 5xFAD mice, we see the generation of these plaques in green, and you see the microglia all around the plaques. And in a TREM2 deficient mice, you see that there's a defect of microglia clustering around the plaques. And so the plaques are free to spread out. And when we look at the morphology of the plaques, you see that in 5-XFAD mice, plaques are more compact. In the TREM2 deficient mice, plaques are more diffuse and tend to spread into the parenchyma. They're more damaging. And indeed, when we look at axonal damage, axonal swelling around the plaques, that you can see here by staining with N-terminus APP, you can see that around the plaque in the 5-XFAD, we see some axonal swelling in green. But in TREM2 knockout mice, we see much more axonal swelling around the plaques. You see the plaque is more ramified. And we saw exactly the same defects in microglia responses, in changes in plaque density, and also in axonal swelling in 5-XFAD mice that were carrying the R47H variant, the human variant, associated with Alzheimer's disease. So we also noticed that TREM2 is essential to sustain the transcriptome changes in response to beta plaques. In collaboration with Ido Amit, several years ago, we've shown that during accumulation of beta plaques, microglia progressively differentiate into different types of activated microglia. A major type of activated microglia is what we call disease-associated microglial dam, which expresses genes involving phagocytosis, lysosomal activity, lipid metabolism, adhesion, and tissue repair. But we also have microglia that become proliferative, microglia that express very high level of MHC class II, microglia that express interferon responsive genes. So we look at this different trajectory of differentiation of microglia in mice with a different genotype, specifically in mice that carry the common variants of human TREM2, which is not associated with Alzheimer's disease, mice that carry the R47H variant of human TREM2, which is associated with Alzheimer's disease, and mice that are knockout for TREM2. And what we see is that in mice with a common variant, we see that all the four trajectories are abundantly represented in these mice. But when we look in mice that have TREM2 deficiency or a defect or a mutation of R47H, we see a significant reduction of all these trajectories. So we ask why TREM2 is important for responsive to beta plaques, and this work was done by Shutan Wong in the lab. We know that TREM2 signals through DAP12. DAP12 is an adapter that contains ITAM motifs in the cytosomic domain. These ITAMs recruit the protein tyrosine kinase SYK, and SYK will then activate a number of downstream signaling events, particularly activation of PI3 kinase, activation of PLC gamma, and also activation of actin cytoskeleton. We also noticed that in 5-XFAD mice, in microglia of 5-XFAD mice, SYK is phosphorylated, which is a sign of activation, whereas it's barely phosphorylated in mice and wild-type mice, suggesting that indeed SYK is activating in the response to amyloid plaques. So we generated mice that lack SYK, specifically microglia, and we crossed these mice onto 5-XFAD mice. And basically, we saw the same result that we observed with TREM2 knockout mice. Again, we see that we have plaques that tend to enlarge, and we see very little microglia around the plaque. Microglia are more distant, and they're unable to respond to the plaques. Now, of course, this results in an increased number and increased size of plaques over time, as you can see here. And in data I'm now going to show you, we also demonstrated that deficiency of SYK in microglia and the accumulation of A-beta plaques in these mice is associated with impairment of memory in behavioral tests and also in anxiety of these mice. So we're particularly interested in looking at one pathway downstream of SYK, the PI3 kinase AKT pathway, because this pathway activates GSK3 beta, which controls cell proliferation, and we know the microglia is proliferating in response to A-beta plaques. And also we were interested in this pathway because we know that AKT is phosphorylated and activated a number of targets on the mTOR pathway, which is important to sustain protein synthesis in cells. And indeed, when we look at phosphorylation of AKT in microglia carrying a defect of SYK, in 5-XAPD mice, we see a defect of phosphorylation of AKT, we see a defect of phosphorylation of JSK3 beta, and we see a defect of phosphorylation of two targets, Estes kinase and NDRG1, which are both targets of the mTOR pathway. So based on this data, we conclude that during neurodegeneration, during Alzheimer's disease, but perhaps more also in other forms of neurodegeneration, a number of different molecular patterns are released that will stimulate different receptors on microglia, toll-like receptor, node-like receptor, scavenger receptor, TREM receptor, and this will lead to microglia activation and the acquisition of different signature, different trajectories that they are responsible for activation and the protective role of Alzheimer's disease. However, this pathway needs to be sustained by TREM2, DAP12, and SYK pathway through mTOR activation. In the absence of this pathway, there's no mTOR activation, there's limited mTOR activation, and therefore a microglia response to beta plaque cannot be sustained. Microglia is not fit enough to respond to A beta plaques. So based on this experiment, we and others have postulated that perhaps we can activate TREM2 during A beta, during Alzheimer's disease, to reduce it, to attenuate the pathology. And we've done several collaborations with different companies. They have developed specific monoclonal antibody against TREM2 and also small molecules. I'm just going to show you one experiment in the interest of time. Here we look at one antibody that is for human TREM2. We tested this antibody on the common variant and the L47H variant, so it stains both variants. We also developed a reporter assay in which TREM2 engagement activates NFAT pathway in a reporter cell line in which NFAT drives the expression of GFP. And so when TREM2 is engaged by an antibody, by a surrogate, the active surrogate ligand, cells will become fluorescent. And as you can see, when we challenge the reporter cell line to express the common variant of human TREM2, we see activation of the reporter cell line, and we also see activation of the reporter cell lines in reporter cells that express the R47H variant. So we asked whether anti-TREM2 can activate microglia in vivo. We generated transgenic mice that express the common variant, the R47H variant of human TREM2, cross them onto TREM2 knockout mice to eliminate endogenous TREM2, and then cross them onto 5-XAPD mice to induce generation of amyloid plaques. We take mice when they develop amyloid plaques at 5 months, and then we inject them intraperitoneally with high doses of antibodies once a week for 3 months, and then at the end of this period, we look at the pathology and the behavior. So I'm just going to show you one piece of data in the interest of time. Here we look at axonal swelling around the plaques. In blue, you see the plaques. In red, you see the axonal swelling. These are mice that treated with control antibodies, and these are mice treated with the anti-TREM2 agonistic antibody. We see a reduction of axonal swelling around the plaques. And we also look at the behavior in these mice, and we saw that indeed agonistic antibodies, anti-human TREM2, reduce abnormal behavioral traits. So a version of this antibody is now completed in phase 1 clinical trial and is currently in phase 2 clinical trial for Alzheimer's disease. I also have to point out that we perform single nuclear RNA-seq in human specimens with Alzheimer's disease. And when we look at the clusters of nuclei that we see, we can clearly identify the cluster of microglia that expresses CSF1 receptor. And when we look at the expression TREM2 and SYK in brain nuclei, we see expression of SYK and TREM2 only in microglia, suggesting that TREM2 and SYK can both be targeted in microglia to activate microglia responses to A beta. And we have shown in other data that we published, and therefore I'm not going to reiterate those, that other receptors, for example, CLEC7A, that can engage directly STIC, can also have a similar effect of TREM2 antibody. So TREM2, of course, is an activating molecule that activates responses in myeloid cells in general, particularly in microglia. There's a number of different activating receptors, but we have to remember that microglia also express inhibitor receptor. And this inhibitor receptor actually will reduce the activity of microglia and also the response to microglial beta plaques. Typically, CD33 is an inhibitor receptor, and mutation, gain of function mutation of CD33 have been shown to be associated with increased risk of Alzheimer's disease, suggesting that inhibitor receptor can also have a role in the pathogenesis of the disease and perhaps can be harnessed to treat Alzheimer's disease. And so we were particularly interested in a family of inhibitor receptors called LIRs that my lab cloned more than 20 years ago. This family of receptor contains 5 different inhibitor receptors, called LIRB1, LIRB2, LIRB3, LIRB4, and LIRB5. There's only one homologue in a mouse called PRB. These receptors have long cytoplasmic tail, which contains tyrosine-based inhibitory motif, or ITIMs, which recur SHIP1 and SHIP2, which dephosphorylates most of the targets that are phosphorylated by STIC. These receptors are encoded on human chromosome 19 by 2 clusters of genes. And so we wanted to see whether this receptor can actually regulate responses to A beta. And this work was done by Jin-Chao Hu and Yun Chen. We were particularly interested in this receptor called LIRB4 or ILT3. LIRB4 is expressed in monocytes, in dendritic cells, in macrophages, plasmacytoid DC, and in rare population of B cells. Now, this receptor attracted our attention also because work by Alex Zhang at UT Southwestern have shown several years ago that LIRB4 is also expressed by acute myeloid leukemia cells, and somehow this receptor interacts with ApoE, and this interaction creates a tumor microenvironment that facilitates the spreading of leukemia. So essentially, this is a receptor for ApoE, although it can also bind to other molecules. For example, it can also bind to fibronectin. And other ligands have been also identified, and we're actually in the process to understand why all these ligands bind to LIRB4, whether they're different in their binding affinity, whether they're different in their effect on signaling properties of LIRB4. But the point here is that LIRB4 is highly expressed in microglia in the brain and is strongly upregulated in Alzheimer's disease. In fact, if we look at all the LIRB and all the LIR-A genes that we looked at, LIRB4 is the only one that is clearly upregulated during Alzheimer's disease. We also found an association between the expression of LIRB4 and the stage of the disease, of Alzheimer's disease. And also there's a correlation between the expression of LIRB4 and the amount of tau that can be found in brain lysates of patients with Alzheimer's disease. We know that microglia, upon activation, is producing ApoE. And indeed, we find that during Alzheimer's disease, the increased production of ApoE, ApoE actually correlates with the stage of the disease. And we notice that the level of ApoE in microglia also correlates with the level of LIRB4. Finally, when we look at the brain section of people with Alzheimer's disease, we can clearly see that, and this is a picture that I showed you before, in which you see the plaques here, you see the ApoE in the plaque, and then you see the microglia around the plaques. And we notice that a large number of these microglia also express LIRB4. So, as I mentioned, LIRs are expressed in human. In mouse, we only have a one peer molecule. There's also two LIR-B molecules that have been reported in mice, LIR-B4B and LIR-B4A. I want to point out that these molecules are in a different chromosomal location, which is not the ortholog location of human LIRs, and therefore the regulation is different. In fact, these molecules are expressed in NK cells, mast cells, T cells, and other cells as well. So we don't think that in mouse, LIR-B4A and LIR-B4B are exactly the equivalent of LIR-4B in human. So because of this, we decided to generate a transgenic mouse model with a back clone that expresses the whole telomeric cluster of LIRs, which includes LIR-B4. And then we took these mice and we crossed them onto a 5D model of Alzheimer's disease. So this is just to show you that mice, we call these mice ILT telo mice. And so these mice express LIR-B4 at very high level, but when we cross them onto a 5xFAD mice, we see an increased expression of LIR-B4 in microglia. Again, this is a quantification. You can see expression in non-5xFAD mice, transgenic mice, but you see that the expression is upregulated in the presence of 5xFAD, and there's a difference here between males and females, and the reason is because female mice accumulate A beta faster and in higher amounts than male mice. And so again, this suggests that the expression LIR-B4 in microglia is correlate directly with the amount of amyloid plaques. By imaging, we can see in these transgenic mice that the plaques are surrounded by microglia, and this microglia expresses high level of LIR-B4. So does LIR-B4 impact A beta accumulation? And so to address this question, we made an antibody specific for LIR-B4 and look whether this antibody has any impact on the accumulation of A beta. So we actually generated an antibody, anti-LIR-B4, it's called ZM301. We mutated the Fc portion of this antibody to prevent any effector function, particular ADCC or ADCP, and then we demonstrated the antibody is actually still capable of binding LIR-B4. And now we take 4 or 5 months old mice that develop A beta plaques, and then we start a weekly treatment, systemic treatment with high doses of anti-LIR-B4 antibody for several weeks. And at the end, we look at behavior, we look at constant, and then we look in the brain lysate as a concentration of antibody, and we also look at the pathology. So what you can see is that, of course, we see very high level of antibodies in the plasma of these mice, but we also see a decent concentration of antibody in the brain lysate, suggesting that our treatment is sufficient to allow the antibody to get into to pass through blood-brain barrier and get into the brain. And so now we look at A beta pathology by staining with methoxyl-4 or 6010 monoclonal antibody, and we see that there's a clear reduction of A beta in treated mice in different areas of the brain, the cortex, hippocampus, and amygdala. And we also done biochemistry. We isolated A beta from the brain lysate of these mice. We look at soluble A beta, and we also look at insoluble A beta. And in both cases, we see a reduction of A beta 42, which is the most toxic form of A beta. So what is the mechanism? We did RNA-seq analysis of microglia, and what we find is that when mice are treated with the anti-LIR-B4 antibody, we see an increase of molecules that are known to be involved in phagocytosis. We see an increase of expression C74, and we see a very strong downregulation of genes that are known to be induced by interferon, and we've confirmed this by gene ontology analysis and also by enrichment analysis. You see again genes that are involved in ROGTPA cycle, which is important for rearrangement, acting cytoskeleton. We see a reduction of an increase of genes involved in Fc receptor dependent phagocytosis, and we see a reduction of interferon response genes. So we wanted to demonstrate directly that the antibody can increase phagocytosis. So to do this, we inject mice with antibodies for several days, and then 3 hours prior to sacrifice, we inject them with methoxyl-4, which will stain A-beta plaques in microglia. We isolate microglia, and then we look at methoxyl-4 positive fibrils in microglia, and we see that when we treat them with anti-LILRB4, there's an increase of uptake of methoxyl-4 positive fibrils. So finally, what is binding to human LILRB4 in vivo, in mice? As I mentioned, one of the ligands of LILRB4 is ApoE. We confirm that that LILRB4 binds also to mouse ApoE, and it binds to both non-lipidated recombinant ApoE, also native ApoE that is being purified from the serum from the plasma of mice. So it binds to both lipidated and non-lipidated forms of ApoE. We also perform our reporter assay, in which LILRB4 is a chimeric molecule that activates the reporter, and when we engage with ApoE, we do see a dose-dependent response of LILRB4 to ApoE, mouse ApoE. Most importantly, when we perform this assay, this reporter assay in the presence of anti-LILRB4 antibody, we block the activation of the reporter, suggesting that the antibody that we generated is an antagonistic antibody. So finally, we did some prediction studies on how LILRB4 can bind to ApoE. And as I mentioned, LILRB4 is a member of the immunoglobulin superfamily, has 2 immunoglobulin domains that form this elbow. And we found that between the domain 1 and domain 2, there's this loop, and this loop is actually in contact with ApoE in all the prediction that we have tested. In addition, we have made mutation of these loops, and we've shown that if we change this loop onto a loop that maintains the structure of LILRB4 but loses the binding feature, LILRB4 is no longer capable of binding ApoE. Now, we confirmed this model by cryo-EM and also by mutation, as I mentioned, particularly the loop swap. Importantly, in structural studies that I'm not going to show you here in the interest of time, we've shown that anti-LILRB4 antibody binds to the same loop that is responsible for binding ApoE, suggesting that anti-LILRB4 prevents the binding to ApoE. So, in summary, what I've shown you is that human LILRB4 is highly expressed in A-beta plaques associated with microglia. The expression correlates with amyloid pathology. That anti-LILRB4 reduces the A-beta burden in our transgenic mice crossed to 5xFAD mice. We particularly, the antagonistic antibody augments the phagocytic activity of A-beta, reduces interferon signature, and this antibody blocks the interaction of LILRB4 with ApoE. We presume that this interaction occurs particularly in A-beta plaques because A-beta plaques are enriched of ApoE, particularly non-lipidated ApoE, and therefore this interaction between ApoE in the plaques and LILRB4 in microglia can strongly affect the response of microglia. And so, obviously, we think that therapeutic activation of microglia with monoclonal antibodies that block LILRB4 or other inhibitor receptor can be equally effective as therapy based on activation of TREM2 or activating receptor. So in the last few minutes that I'd like to go back to TREM2 and move away from our work on neuropathology and tell you about our work in other areas. Now, we know that TREM2 has been implicated in adipose tissue macrophages, in liver macrophages, in foamy macrophages of atherosclerosis. And there's a number of different studies being published in this topic, but we're particularly interested in the expression of TREM2 in tumors. And this for two reasons. The first reason is that we noticed that TREM2 is highly expressed in infiltrating macrophages in a number of different human tumors. Basically, we've looked at more than 200 different tumors, and we always find infiltration of TREM2 positive macrophages in human. And in addition, we noticed that the high level of expression of TREM2 in tumors correlate with a negative prognosis in two types of cancer, in a colorectal carcinoma and triple-negative breast cancer, in a TCGA database of NIH. So we know, of course, that tumor-associated macrophages in general have an immunosuppressive function. They reduce T-cell activation, they induce Tregs, they produce a number of anti-inflammatory and immunosuppressive cytokines. They also release metabolites that reduce the activation of T-cells and in general immune responses. And so we ask the question whether we can block TREM2 in these settings and increase the immunogenic activity of macrophages in the tissue. And this work was done by Martina Mulgore, Jin-Chao Hu, and Yun Chen. Martina will be soon starting her work as an independent investigator in Tampa and at the Moffitt Cancer Center. And so I'm sure we'll see more work coming from her lab soon. So in this preliminary experiment, we took 3 different types of tumors. This MCA derived sarcoma, which was developed by Bob Schreiber, MC38, the PYMT model of breast cancer, and then we injected subcutaneously in wild-type and TREM2 knockout mice. We saw a reduction of the growth, not dramatic, but it was very consistent. And Jelani actually recently showed that this is also the case in a model of prostate cancer. So we looked at the macrophages in these tumors in the 2 different genotypes. So first of all, you can see that there's different macrophages infiltrating. This is the MCA sarcoma tumor. And many of these, I would say most of these express TREM2, although at different levels. And when we look at this cluster of macrophages across the 2 different genotypes, we can see that in a TREM2 knockout mice, we see a reduction of CX3CL1 macrophages. We also see a reduction of CD206 macrophages, which are always associated with immunosuppression. And we see also an increase of CXL9 positive, CD83 positive macrophages, which are more associated with immunostimulation. And when we also look at T cells by single cell RNA-seq, we see an increase of CD8 cells that produce interferon gamma and express PD-1, which we confirm also by flow cytometry. So, as I mentioned, the data were very consistent, but was not dramatic. So we wanted to see whether we can harness this effect to enhance checkpoint blockade. And so we used, again, our MCA model of carcinoma. Now, this model is of a sarcoma. This model is highly susceptible to anti-PD-1. In fact, if we use optimal concentration, optimal treatment, anti-PD-1 will completely eliminate the tumor. So to mimic PD-1 resistance, we use a suboptimal treatment with anti-PD-1, in which we delay the injection PD-1 when the tumor is already growing, day 8, day 9, day 10. And if we do this, then the anti-PD-1 no longer eliminates the tumor, but only blocks the growth of the tumor and inducing tumor dormancy. So we perform a similar experiment in TREM2 knockout mice. So here are the wild-type mice, the TREM2 knockout mice. There's the delay of growth. In anti-PD-1 suboptimal, we see tumor dormancy, and we see complete elimination of the tumor with with TREM2 deficiency plus suboptimal anti-PD-1. And we obtained similar data in MC38 colorectal carcinoma. So we wanted to see whether we can recapitulate this with anti-TREM2 monoclonal antibody blockade. Jin Xiaohu made an antibody, anti-TREM2. We made a recombinant antibody that is effectorless, and so it does not engage the Fc portion. It does not induce ADCC. It does not induce ADCP. And we also tested whether this antibody is an antagonistic antibody. So again, this is our reported assay in which engagement of TREM2 is induced in GFP. We engage TREM2 with a lipid ligand, for example, HDL. We see activation of reporter, but in the presence of our monoclonal antibody, anti-TREM2, we see a reduction of activation of the reporter, suggesting that the antibody can act as an antagonist. So now we do our experiment with the anti-TREM2 antibody. The anti-TREM2 antibody reduces tumor growth. The suboptimal anti-PD-1 is inducing tumor dormancy, and the anti-PD-1 plus anti-TREM2 antibody induces elimination of a tumor. We look at the macrophage at the single cell level in these tumors, in this different treatment. And again, we see a number of different cluster of macrophages. More or less, most of these macrophages express TREM2 at different levels. And when we look at mice that have been treated with anti-TREM2 plus anti-PD-1 or anti-TREM2 alone, we see a clear reduction of CD206 positive macrophages, which are immunosuppressive. We see a reduction of proliferating macrophages, and we see an increase of macrophages express a nitric oxide synthase 2, or inducible nitric oxide, which is usually associated with immunostimulation. And again, these changes in macrophage landscape are associated with an increase of CD8 T cells that produce interferon gamma, and also an increase of CD4 T cells that produce TNF alpha. So what I've shown you is that TREM2 is expressed in tumor-associated macrophages, that a high level of expression is negatively correlated with survival in triple-negative breast cancer, colorectal carcinoma, that TREM2 deficiency and blockade of TREM2 with a specific monoclonal antibody reduces tumor growth and enhances the effect of anti-PD-1. And in general, we think the anti-TREM2 treatment is sufficient to skew the myeloid compartment towards a more pro-inflammatory, pro-activating environment. And I have to say that some of these experiments have also been confirmed by other studies, particularly by Ido Amit and by Max Schrammel, and also by Miriam Meral. So finally, in the last few minutes, I think I have 5 or 6 minutes left. I'd like to tell you our recent work on how TREM2 deficiency can improve anti-PD-1 efficacy. And this is work that was done by Blanda De Lucia together with Martina and Daria Kantakova in collaboration with Jeff Gordon. So in our experiments, we take wild type and knockout mice. We co-house them from birth to reduce, to attenuate changes in differences in microbiota. We inject them with tumor, and then we separate them by genotype. We inject them with anti-PD-1, and then we look at the tumor growth. And this is what we see. We see that in TREM2 deficiency, we see less tumor growth. With anti-PD-1, we see tumor dormancy. And with anti-PD-1 plus TREM2 deficiency, we see reduction. We see tumor elimination. We also done this experiment in which we took mice, co-house, TREM2 knockout and wild type, and we co-house them throughout the experiment, also during anti-PD-1 treatment. And if we do this, we still see a difference between wild type and TREM2 knockout mice in tumor growth, but we no longer see the synergistic effect of TREM2 deficiency with anti-PD-1. So this was surprising to us, of course, and we didn't see also changes in the T cell compartment. So obviously this implicated a potential effect of microbiota, and so we performed fecal microbiota transplantation experiment in which we take our TREM2 and wild type mice, we take the sickle content prior to injection of a tumor. In a different group, we inject them with a tumor, we separate them, and then we inject them with anti-PD-1. After the first injection of anti-PD-1, we take the sickle content. And so basically, we have 4 different types of donors. We have donors that are TREM2 deficient and TREM2 knockout prior to injection of anti-PD-1, and we have donors TREM2 and wild type knockout after injection of anti-PD-1. So we take wild type and TREM2 mice, treat them with antibiotic, and then reconstitute them with 4 different donors of sickle material. Then after reconstitution, we inject them with the tumor, we separate them by genotype, and we treat them with anti-PD-1. And then we look at tumor growth. So what we see here is that in wild type mice that have been reconstituted with sickle content of knockout mice at day 0 prior to injection of anti-PD-1, wild type mice do not reject the tumor. But when we take the same wild type mice and then we reconstitute them with sickle matter from knockout mice at day 12 after injection of anti-PD-1, the tumor is rejected. Conversely, if we take knockout mice that usually reject the tumor and we reconstitute them with the sickle matter of wild type mice prior to injection of anti-PD-1, the tumor is still rejected. But when we take knockout mice and then we reconstitute them with sickle matter of wild type mice at day 12 after injection of anti-PD-1, the tumor is no longer rejected. Now, we also look at the effect of several antibiotics. So here we look at the tumor rejection by TREM2 deficient and TREM2 wild type mice. Treated with vehicle, TREM2 deficient mice reject the tumor, wild type mice don't. But if we treat them with ampicillin, they don't reject the tumor. If we treat them with vancomycin, they don't reject the tumor. If we treat them with metronidazole, they still reject the tumor, suggesting that the microbial species that are responsible for the rejection are gram-positive. So we did a number of studies to identify the species that are responsible. They're different in the microbiota of these different mice, separated at 0, separated or co-housed, treated with different antibodies. I'm not going to detail of this. What I want to show you is simply that at the end of this analysis, we saw a clear difference in the mice treated anti-PD-1 TREM2 deficient, and this was the abundance of a species called Ruminococcus nevus, or nevus. R. nevus has been previously identified in Crohn's disease, particularly in patients with flares of inflammation. And it has been suggested that inflammation actually facilitates colonization of R. nevus because inflammation is associated with reduction of oxygen tension, and R. nevus is an anaerobic bacteria. And so this could be one of the reasons why we see an expansion. And so we ask whether the treatment of anti-PD-1 in a TREM2 deficient mice is inducing some sort of gut inflammation. Now, this is not the case. We don't see a clear clinical colitis in TREM2 mice treated with anti-PD-1, but we do see increase of cytokines like IL-17, TNF-alpha, reduction of IL-10. And so we think that there's a subclinical inflammation. And perhaps the subclinical inflammation is mediated by TREM2 positive intestinal macrophages that can contribute to inflammation. In fact, what we did here was to take mice, inject them with a tumor, inject them anti-PD-1, and then perform single-cell RNA-seq of macrophages in the gut of treated mice. And what you can see is that when we take TREM2 deficient mice and we treat an anti-PD-1, we see changes in the abundance of certain cluster of macrophages in the gut. And we see particularly an increase of this cluster 0 and cluster 2 and a reduction of cluster 1. Now, cluster 0 expresses pro-inflammatory chemokines, CCL3, CCL4, which is increased. Cluster 2, which is also increased, expresses interleukin-1, NLRP3, CD80. These are also markers of activation. Cluster 1, which is decreased, expresses stabilin-1, CD163, MRC1, CD206, which are markers of reduced inflammation. So we think the anti-PD-1 in TREM2 deficient mice changes also the colonic macrophages toward a pro-inflammatory phenotype. And so these are modeled. We think that during treatment with anti-PD-1 has really no major impact in the gut in the presence of TREM2. However, in the absence of TREM2, macrophages becomes more activated, and they generate a pro-inflammatory environment that facilitates RNAs. And we think that somehow the RNAs bloom is facilitating anti-tumor T-cell responses and reduced tumor growth. And so we ask the question finally whether RNAs can replace TREM2 deficiency and enhance the anti-PD-1. And so here we take wild-type mice, we inject them with a tumor, we gavage them with RNAs for several days, we treat an anti-PD-1, and then we look at tumor growth. And we see that tumor is in the presence of RNAs is eliminated, which we don't see with a control bacteria, for example, Clostridium shingles, which is also anaerobic and also partially expanded in TREM2 deficient mice. So obviously, RNAs has an impact on tumor growth. And so we ask whether it has an impact on the tumor infiltrate in the immune infiltrate of the tumor. And so here we perform a same experiment. We take wild-type mice, inject them with a tumor, gavage with RNAs, treat them with anti-PD-1, and then look at the tumor microenvironment, immune microenvironment, both T cells and macrophages. We see an expansion of effector CD4 and CD8 T cells, but we also see very interesting changes in a macrophage compartment. We see that two cluster of macrophages are expanded and one cluster of macrophages is reduced. So we look at genes that are differentially expressed in these macrophages, and what we notice is that in mice that treated with RNAs, we see a significant reduction of clusters of macrophages that express TGF-beta receptor 1. So this we see a reduction of macrophages that are responsive to TGF-beta. And on the other hand, we also see an expansion of certain clones of macrophages that express interferon gamma receptor 1. So these macrophages are more responsive to interferon gamma. And so what we conclude is that the housing condition obviously have an impact on anti-PD-1 response in TREM2 deficient mice. We think that this is because the microbiome from TREM2 deficient mice treated with anti-PD-1 is responsible for enhancing the anti-PD-1. And we've shown that this is happening in wild-type mice, both with sickle transplant and also with RNAs, because RNAs seem to be, at least in our housing facility, seems to be responsible for this rejection. And I've shown that RNAs alone can enhance anti-PD-1 tumor growth, impacting infiltrated T cells and tumor macrophages. So, of course, there are a number of different questions that we need to address. Why RNAs expand in the gut? Is the subclinical inflammation sufficient? But most importantly, is how RNAs can, in the gut, can facilitate T cell responses in tumor. With obviously, bacterial metabolism could be involved, but there could be other mechanisms as well. So I think I mentioned all the people on the talk that are responsible for this work. This is my lab, and I like to acknowledge all of them. And we also have important collaborations. I like particularly to mention Yoni Kipnis and Dave Holzman for our work on Alzheimer's disease, Bob Schreiber and Jeff Gordon for our work on tumors. And we have important collaboration with brain banks that provide us with samples for human. We collaborate with Ido Amit on single-cell RNA-seq. We have industrial collaboration to study the agonist of TREM2, antagonist of TREM2. And then also I'd like to acknowledge Luigi Poliani, William Vermi, and University of Brescia for their help with pathology. So I think I'll stop here. And if there's any time, I'll take any question. Thank you for your attention.
Thank you very much for an amazing talk. I think we have some questions. Jelani, can you handle it?
Yes. So The first question is from Adam Prater. Adam, would you like to unmute and ask your question, or would you like for me to ask it for you?
Oh, that was Adam.
Oh, I'm sorry. JD is the first question. JD, would you like to unmute? The question from JD is to Dr. Colonna. Can you comment on approaches targeting TREM2 and LIL receptor in cancer and its implications in neurotoxicity?
Also, okay, so the question is whether the use of these antibodies in cancer can have any neurotoxicity. Is that correct? So the answer is probably not, because systemic injection of antibodies has a very poor yield in crossing the blood-brain barrier. So for the dose of antibodies that we use in treatment of tumors, at least in mice, these doses are not sufficient to cross the blood-brain barrier and reach very significant concentration of brain. In the brain, for the brain, we need to use very high concentration, let's say between 60 and 100 milligrams per kg. So these are very high doses that we need to inject for crossing blood-brain barrier. So I don't think there's any problem with neurotoxicity. There's clinical studies ongoing, clinical trials, so we'll see. But they haven't reported, at least in phase 1 clinical trial, nobody has reported any neurotoxic effect.
So could I ask a follow-up question? When you inject these high doses of agonistic antibodies, do you see any toxicity, like liver toxicity? Do the Kupffer cells express TREM2?
No, so let me say that TREM2 is expressed in many macrophages,
right?
But the level of expression in general is very low. So it's only upregulated in certain conditions. For example, in Alzheimer's disease, you have a very strong upregulation around the plaques, because away from the plaques, you don't see much of a upregulation of TREM2. So we personally haven't done any study in the liver, but we think that in the liver also, TREM2 is upregulated in liver steatosis, in fatty liver disorder, and the same also is true in adipose tissue. You see high expression of TREM2 in adipose tissue macrophages after high-fat diet. So in this condition, maybe there could be some toxicity, but in the absence of this condition, the level of TREM2 is not highly expressed. So I don't think that the antibody will have much of a toxicity in that context.
Okay, one more quick question. Then in those settings, steatosis and upregulation in adipose tissue, would inhibiting TREM2 be beneficial then? Would you predict?
So it's so we don't work on that. So I can only I mean, we would like to do that, but we only have a certain bandwidth. So there's some discrepancy on the data on adipose tissue. There's one paper that shows that yes, TREM2 blockade has an impact on TREM2 deficiency, at least in TREM2 knockout mice. There's an amelioration of metabolism, and there's a study that shows no effect. So I think it's still unclear. You know, as you know, in high-fat diet experiment, there's a lot of changes also occurring in the microbiota. And now we know from our studies that TREM2 has an impact on microbiota. So that I think the whole story has to be revisited. And again, you know, in liver, we see sometimes there being reported the opposite effect. So I think it's still an open question. And in fact, I can mention that Mira has also shown some data that in general, trying to blockade is beneficial in tumor, but in the liver, things may be different. So the effect of TREM2 may be context dependent, depending on the effect of macrophages, really.
Yeah. Okay. Thanks.
You have two questions from Santiago Bedoya. Santiago, would you like to unmute and ask, or I can ask? I can read from the text.
Sure, thank you. I'm gonna have to log off soon, so I'm gonna ask the second one. But in terms of the anti-PD-1 treatment and the TREM2 knockout and the wild type, so you said that when you co-house them together under treatment of anti-PD-1, you actually lose the effect So that to me suggests that anti-PD-1 treatment in the wild-type mice is actually somehow inducing a negative bacteria that can counteract the R navis in the knockout. So I was curious if, or maybe you have another idea of why that might be knocking it out, because in that case, you should still have the R navis inducing in the knockout, right?
So, as I say, this is one of the questions that we're asking is why we have an expansion of our neighbors, right? So one idea that we have is that if you let me tell you really our hypothesis. Our hypothesis is that anti-PD-1 on T cells will upregulate molecules like CD40 ligand that will activate macrophages. There's plus CD40. In general, this activation may not be very strong, but if you in the absence of TREM2, you may have a stronger activation of macrophages. This may lead to some pro-inflammatory situation in which you facilitate the expansion of our neighbors because maybe you reduce the tension of oxygen. Now, having said that, we're not even sure if our neighbors per se is responsible of what we see, because when you see our neighbors may have an effect on the whole ecosystem of the gut, right? So when you increase a certain bacterial species, you can also reduce other bacterial species and vice versa. So, the gavage experiment will point the fingers to our neighbors, but nevertheless, we cannot exclude that our neighbors may have an effect on other bacteria that may have a immunosuppressive effect. So we're still not, I think it's interesting that we find our neighbors as one bacteria that is responsible. We don't think our neighbors is the only bacteria that can do that. There's probably other bacteria. But in our facility, our neighbors seems to have this effect. And so we're trying to understand also how our neighbors is actually working.
Awesome. You have thank you for that question and for that great answer, Dr. Colonna. You have a question from Alex Lee. Alex, would you like to unmute?
Hi, yeah, great talk. Thanks for that. I just have a really quick question about R navis. I'm just curious if anyone's looked at the gut microbiome of like anti-PD-1 responders, and if anyone has noticed a presence of like R navis in those patients.
Yes. So so far nothing has been reported in human. We know from work of George Strinkieri and others that the gut microbiota may have an impact on the response to anti-PD-1. There are studies that will show you that in patients, that there's an expansion of certain species in patients that respond to the anti-PD-1, and then the fecal transplant from donors that are responsive to anti-PD-1 has an impact on the effectiveness of anti-PD-1 in patients that are not responsive. And I heard this data from Jennifer Vargo also. I haven't seen the published data yet, but this has been presented in a few meetings in which our neighbors has been implicated or has been actually found also in patients. But I haven't seen the published data, so I cannot comment on that. But I think we'll probably see our neighbors coming up also in some human studies. But I want to emphasize that we don't think that our neighbors is the only species that can do that. That's why we're trying to see whether metabolites produced by our neighbors are responsible for what we see, because same metabolites may be produced also by other species as well.
Really fascinating. Thanks.
Thank you.
Api Mitra has their hand up.
Thank you. Excellent talk, Dr. Colonna. Just a couple of questions I have. Like, one of the aspects is the TREM2/6 pathway. When it's getting activated, neurodegeneration disease versus your cancer space. Did you see cytokine chemokine biology dynamics are similar or they're diverse? One of the questions is that.
Well, I'm sorry, the question is whether TREM2 is inducing cytokines?
Yeah, I mean, the TREM2 activation with the SIG pathway, did you see cytokine chemokine in those microglia versus in the tumor space,
Oh.
like say macrophage or other myeloid population, did you see the cytokine dynamics or chemokine dynamics are different or they are correlated?
Good question. I think it's different. We see different effects.
Mm-hmm.
The reason, my view of TREM2 is some sort of co-stimulatory molecule. So it's a molecule that supports the response of cells to a certain stimuli. So in the neurodegenerative model that we use, mice are microglia are responsive to protein aggregates. Protein aggregation does not induce a pro-inflammatory response. In fact, we don't see in these mice, in 5-XFAD mice, we don't see production of interleukin-1, interleukin-6, TNF-alpha, nothing like that. We only stimulate a phagocytic activity, mostly activated phagocytic activity. And so what you see is that TREM2 is supporting the phagocytic activity of these cells. In a tumor, you probably have a different stimulus that activate tumor-associated macrophages. This could be, and depends on the tumor or model that's used, but for example, apoptotic cells or lipoproteins. And so in this case, you have a different response. The TREM2 will support that particular response. Okay, so I think that the response that you see in different tumors may be different in different contexts.
And another question I have on, because there are several checkpoint markers also expressed on the myeloid cells like PD-L1, TIM3. And so my question is, how do you give a preferential? I mean, like a TREM2, is the superior compared to others, or do you think they are also equally relevant in cancer immunotherapy space?
So, well, we think that, so the advantage of TREM2 is that TREM2 is highly expressed in a pathology, right? And for example, in neurodegeneration, TREM2 is only expressed around the plaques. So we think that the antibody may have a very selective effect on cells that are directly involved in pathology. Having said that, for example, LIR-B4 is also highly expressed, for example, in neurodegeneration, only around the plaques. You don't see LIR-B4 or see very little LIR-B4 away from the plaques. So in that respect, maybe blocking anti-TREM2 or activating anti-TREM2 or blocking LIR-B4 may have similar effect, may be equivalent. And instead that, we also noticed that the response, microglia response to activation of TREM2 and blockade of LIR-B4 are not entirely overlapping. So we're trying to understand whether signaling pathways are, in our simplified view, is a yin-yang, right, of item, item. But in fact, the signaling can be slightly different.
And last question I have on the combination therapy of the Arnivis with the anti-PD-1. Did you see more the generation of clonogenic T cells, or some specific clones are getting activated, and how the DC microbiology, like conventional DC type 1s, how they are modulated in that scenario?
Yeah, good question. We haven't looked at the clonality of T cells yet, although we have, in the MCA sarcoma model that we use, Bob Schreiber has defined the antigen, so we'll be able to trace the specificity of this of T cells using tetramers. And instead that, I'm not entirely convinced that the response that we see in a tumor is specific. There could be some, as I said, metabolites produced by bacteria may have a systemic effect and may facilitate more recruitment of these cells in the tumor bed, or may somehow facilitate the pre-existing T cell, tumor-specific T cells in the tumor. But we really don't have a clear question to that, and certainly that's a very important question.
Thank you.
The next question,
Marco, do you have time for a few more questions? Or... Marco?
Yeah, of course.
Okay,
yeah.
okay. All right, go ahead, John.
So the next question is from Myeloid Network founder, the great Johanna Joyce. Would you like to unmute? Johanna?
Hi, yes, thank you.
Hi.
Marco, wow, that was such an incredible talk, so much to digest there. It was just amazing. I will try to limit myself in the questions that I ask you. The question I put in the chat was just to know if you see different effects in terms of the microbiome looking at the TREM2 knockouts, where of course their microbiome will be altered throughout their whole, lifespan, versus looking in the context of TREM2 inhibition,
Yes,
which is of course a defined treatment window.
it's a very good question. It's a very good question. So we're trying to address that. We're actually, we have submitted the paper and somebody asked the question.
It wasn't
But-
me.
It's a tough question because we have to repeat all the experiments using a specific monoclonal antibody. But the bottom line is that I expect this to be different. Because, having said that, so there's two points I'd like to make. The first point is that probably it'll be different because, as I mentioned, the TREM2 blockade has is short term, and so it may have different effect, right? So it may still be a different mechanism. Having said that, in a TREM2, if you compare the TREM2 knockout mice with wild-type mice prior to the tumor experiment, we don't see much of a difference in the microbiota without anti-PD-1, okay? But, I cannot exclude that there's minor changes in the microbiota that we amplify with the anti-PD-1, and we have not been able to detect early on just because they're below the resolution of our techniques. So there could be, it could well be. It could well be.
Yeah, it does seem that the PD-1 treatment exposes that differential.
Absolutely.
If I can ask a second question, if I may,
Sure.
it relates to the nature of the bacterial species. You talked a lot about the bacterial metabolites, of course. I just want to ask your opinion on whether you think, and it probably can be yes, right? That depending on the tumor type, there will of course be a different interplay with the gut microbiome, with the bacterial microbiome, maybe within the tumor, for example. And then this particular species or genus that you identified may or may not be relevant depending on the tumor type. So is that something that you've looked at all?
You're totally totally right. And the truth is that we don't know exactly how RNA works, right? So as you mentioned, so one of the ideas that we have is that may release metabolites. But the other possibility is that also some of these RNA migrate goes into the tumor.
Exactly.
We don't have any evidence for that, but it's not easy to find it. Okay, it's not easy to demonstrate that. The other possibility is that somehow, and this is something that we tested, or our results are negative, but it doesn't mean that it doesn't happen. Is that you elicit T cell responses in the gut, and then these T cells migrate into the tumor and has a tumor effect, right? So there's some sort of molecular mimicry. And we actually done some experiment that we've shown that T cells actually migrate from the gut into the tumor, but we also see T cells that migrate into the tumor that are not derived from the gut. So it seems to be more systemic effect. But as you say, sure, in certain tumor types, certain bacterial species may be important. In others, may be completely irrelevant, or maybe even detrimental. So certainly something that we should expand our panel of tumors that we looked at with RNA and see whether these RNA is important in all the tumors that we looked at. And also, we should expand it also with different checkpoint blockades, right? Because
exactly. Yeah.
But it could be very different to anti-CTLA-4 or anti-PD-L1.
Yes. Wonderful. Thank you. Thanks again.
Thank you.
Yeah, final two questions, Dr. Colonna. One is from Motion. Motion, would you like to unmute if you're still here?
Hi, thank you, Dr. Colonna, for your nice talk. My question is related to Suber TREM2. Previously, you have reported that TREM2 is involved in differentiation. And do you have any further evidence on the function of TREM2 in tumor?
The function of TREM2 in tumor macrophages. So that's a good question. We know that blockade of TREM2 or defect of TREM2 is changing the landscape of macrophages. We don't know exactly how that works. there's two possibilities, right? The one possibility is that TREM2 elicit certain signaling pathways in all tumors that facilitate inflammation versus or activation versus suppression. The other possibility, so in the past, we have shown that TREM2 can synergize with CSF1 receptor to sustain macrophage survival. We've shown that, I have to say, we've shown that in the past with bone marrow-derived macrophages. So if you starve bone marrow-derived macrophages from CSF1, macrophages survive very well for 48 hours, but if they're TREM2 deficient, actually they undergo apoptosis. They stop proliferation, they undergo apoptosis. So the other possibility is that when you block TREM2, you reduce the responsiveness of macrophages in the tumor to CSF1 stimuli. And some macrophage population may be more susceptible than others to starvation of CSF1 or reduction of CSF1 receptor signaling. And that may explain the relative abundance of different macrophage population within a tumor.
And your final question, Dr. Colonna, is from Shweta Joshi.
Hi, Marco. Very nice talk. And I really like the TREM2 signaling pathway. So my lab also works on SYK signaling, and we found that SYK has a role in macrophage-mediated immunosuppression. And somehow the results I am seeing on cancer models, it's similar when we used our SYK knockout models and SYK inhibitors. It also plays polarizes the macrophages into immunostimulatory phenotype, and we also see an increase in T cells and infiltration of T cells, proliferation of T cells in the tumors. So I would like to know in your single-cell RNA-seq data, or so if you have seen a correlation between TREM2 and SYK in the macrophages. Is SYK also expressed in the specific macrophage phenotypes where TREM2 is expressed? And when you block TREM2, do you see any changes in the phosphorylation of SYK or expression of SYK?
No, we didn't see. It's a very good question, and thank you for asking a question. We didn't see difference in the expression of SYK. Of course, you know, SYK will be activated by TREM2, so you would expect to see more changes in the phosphorylation of SYK, or maybe in downstream signaling of SYK. But it's certainly something that we should look more carefully, especially if we see difference in the target of SYK expression. So we haven't seen that, but I would expect that, you know, as we say, you know, and I'm happy to hear your data. I mean, obviously, blockade of SYK should have a similar effect that we see in a TREM2 knockout. Although the other interesting thing about TREM2 is that TREM2 can signal through SYK, DAP2 SYK, but it can also signal through another pathway called DAP10 PI3 kinase. And so, at least in the central nervous system, there's some discrepancy between the TREM2 signaling, TREM2 knockout, and the SYK knockout in terms of transcriptome. Okay, but you know, what we haven't really done a very good job, but we should do that in looking at SYK as well, and both in the PI3 kinase pathway in both knockout mice.
Yeah. Thank you. So another question, SYK is activated downstream of various receptors. We have found out that it's activated in tumors downstream of alpha-4 beta-1 integrin, Fc gamma receptors. So I would like to know more of your thoughts. I know that TREM2 activates SYK, but would it be good to use SYK inhibitor? Because we saw almost similar effects. It would be interesting to see that the results of TREM2 blockade versus SYK inhibition. Although I know that SYK also has an impact on B cells. We found really very good, interesting results in infiltration of B cells in PDAC tumors. But you know, it would be good to compare that just SYK inhibition alone, because
that's a very good point. You know, we should do that. I don't know if we will be able to do that, because obviously they will require, you know, taking SYK flux mice and look at, you know, so it's a whole interesting pathway. But I heard there's the data on dectin, for example, in tumors. Mm-hmm. So dectin is another receptor that signals through SYK, and so it'd be interesting to see what happens, you know, if we block dectin, or if we activate dectin in a context of tumor, right? So obviously there's different pathway integrins, obviously, can also activate SYK. And so there's, as you mentioned, there's different pathways that may be interesting to explore and compare with TREM2. Absolutely.
Yeah, thank you.
Oh, thank
So,
you.
Marco, I have a comment. So you've showed that the signaling downstream of TREM2 leads to mTOR activation, right?
Yeah.
So we found that mTOR is critical in suppressing the pro-inflammatory response. So if you inhibit mTOR with rapamycin, you can really robustly stimulate pro-inflammatory response. So just a thought is that regulating TREM2 may affect pro-inflammatory signaling because it inhibits mTOR, right?
As I say, we see TREM2 as a molecule that sustains the response of the cell.
Yeah.
But if the cell is producing inflammatory responses, blockade of TREM2 will probably, and reduction in mTOR will probably reduce the support to that particular response. But there's different opinion, right? Some people think there's a phagocytic receptor. We don't see, it binds to lipids, but we don't think is a very strong phagocytic receptor.
Yeah. So in that way, I think it's very similar to the PI3 kinase contributions to macrophages, whether it's gamma or delta, depending upon the cell type, So I think it's very interesting.
So we haven't really look at that, but macrophages usually, you know very well. I mean, there's certain isoforms of PI3 kinase that probably are affected by TREM2, right?
Yeah.
A long time ago, we tried to play with PI3 kinase alpha, P1110 alpha, which can be overexpressed.
Yeah.
Conditionally, but we dropped it because it was too much work. It was too much cross and too much. So we just, unfortunately, we didn't do it. It would have been an interesting experiment.
Yeah, I mean, I think once a PI3 kinase is activated, they'll all do the same thing.
Yeah, presumably, yes, absolutely.
Well, thank you so much for a wonderful talk.
No, thank you. Pleasure to be here, and thank you for the question. Really, a lot of insightful questions and a lot of things to think about it.
Yeah. Well, really, it was wonderful, and we'll send you a link to all the questions in case you want to look at them again. And
absolutely. And if you guys want to send any questions by email, please feel free. I'll try to answer all of them. I'll try.
Great. Well, look forward to seeing you again soon.
Okay.
All right.
All right.
Bye. Thanks.
Take care.
Thanks so much. Bye-bye.
Bye-bye.